An oocyte, paragraph-cyte, ovocyte, or oocyte, is a female gametocyte or germ cell involved in reproduction. In other words, it is an immature ovum, or egg cell. An oocyte is produced in the ovary during female gametogenesis. The female germ cells produce a primordial germ cell, which then undergoes mitosis, forming oogonia. During oogenesis, the oogonia become primary oocytes. Formation. The formation of an oocyte is called oocytogenesis, which is a part of oogenesis. Oogenesis results in the formation of both primary oocytes before birth, and of secondary oocytes after it as part of ovulation. Characteristics. Cytoplasm oocytes are rich in cytoplasm, which contains yolk granules to nourish the cell early in development. Nucleus during the primary oocyte stage of oogenesis, the nucleus is called a germinal vesicle. The only normal human type of secondary oocyte has the 23rd chromosome as 23, X, whereas sperm can have 23, X or 23, Y. Nest the space within an ovum or immature ovum is located as the cell nest. Maternal contributions. Because the fate of an oocyte is to become fertilized and ultimately grow into a fully functioning organism, it must be ready to regulate multiple cellular and developmental processes. The oocyte, a large and complex cell, must be supplied with numerous molecules that will direct the growth of the embryo and control cellular activities. As the oocyte is a product of female gametogenesis, the maternal contribution to the oocyte and consequently the newly fertilized egg is enormous. There are many types of molecules that are maternally supplied to the oocyte, which will direct various activities within the growing zygote. Avoidance of damage to germline DNA the DNA of a cell is vulnerable to the damaging effect of oxidative free radicals produced as byproducts of cellular metabolism. DNA damage occurring in oocytes, if not repaired, can be lethal and result in reduced fecundity and loss of potential progeny. Oocytes are substantially larger than the average somatic cell, and thus considerable metabolic activity is necessary for their provisioning. If this metabolic activity were carried out by the oocyte unregistered trademark S-own metabolic machinery, the oocyte genome would be exposed to the reactive oxidative byproducts generated. Thus it appears that a process evolved to avoid this vulnerability of germline DNA. It was proposed that, in order to avoid damage to the DNA genome of the oocytes, the metabolism contributing to the synthesis of much of the oocyte unregistered trademark S constituents was shifted to other maternal cells that then transferred these constituents to oocytes. Thus, oocytes of many organisms are protected from oxidative DNA damage while storing up a large mass of substances to nurture the zygote in its initial embryonic growth. mRNAs and proteins during the growth of the oocyte, a variety of maternally transcribed messenger RNAs, or mRNAs, are supplied by maternal cells. These mRNAs can be stored in mRNP complexes and be translated at specific time points. They can be localized within a specific region of the cytoplasm, or they can be homogeneously dispersed within the cytoplasm of the entire oocyte. Maternally loaded proteins can also be localized or ubiquitous throughout the cytoplasm. The translated products of the mRNAs and the loaded proteins have multiple functions. From regulation of cellular housekeeping such as cell cycle progression and cellular metabolism, to regulation of developmental processes such as fertilization, activation of zygotic transcription, and formation of body axes. Below are some examples of maternally inherited mRNAs and proteins found in Xenopus laevis oocytes. Mitochondria the oocyte receives mitochondria from maternal cells, which will go on to control embryonic metabolism and apoptotic events. The partitioning of mitochondria is carried out by a system of microtubules that will localize mitochondria throughout the oocyte. In certain organisms, such as mammals, paternal mitochondria brought to the oocyte by the spermatozoon are degraded through the attachment of ubiquitinated proteins. The destruction of paternal mitochondria ensures the strictly maternal inheritance of mitochondria and mitochondrial DNA or mtDNA. Nucleolus In mammals, the nucleolus of the oocyte is derived solely from maternal cells. The nucleolus, a structure found within the nucleus, is the location where RNA is transcribed and assembled into ribosomes. While the nucleolus is dense and inactive in a mature oocyte, it is required for proper development of the embryo. Ribosomes maternal cells also synthesize and contribute a store of ribosomes that are required for the translation of proteins before the zygotic genome is activated. In mammalian oocytes, maternally derived ribosomes and some mRNAs are stored in a structure called cytoplasmic lattices. These cytoplasmic lattices, a network of fibrils, protein, and RNAs, have been observed to increase in density as the number of ribosomes decrease within a growing oocyte. Paternal contributions: the spermatozoon that fertilizes an oocyte will contribute its pronucleus, the other half of the zygotic genome. In some species, the spermatozoon will also contribute a centriole, which will help make up the zygotic centrosome required for the first division. However, in some species, such as in the mouse, the entire centrosome is acquired maternally. Currently under investigation is the possibility of other cytoplasmic contributions made to the embryo by the spermatozoon. During fertilization, the sperm provides three essential parts to the oocyte: a signaling or activating factor, which causes the metabolically dormant oocyte to activate; the haploid paternal genome; the centrosome, which is responsible for maintaining the microtubule system. See Anatomy of sperm. Abnormalities: non-disjunction, a failure of proper homolog separation in meiosis I, or sister chromatid separation in meiosis II can lead to aneuploidy, in which the oocyte has the wrong number of chromosomes, for example 22X or 24X. This is the cause of conditions like Down syndrome and Edwards syndrome in humans. It is more likely with advanced maternal age. Some oocytes have multiple nuclei, although it is thought they never mature. See also, cortical granule, follicular genesis, oocyte maturation inhibitor, polar body, symmetry breaking and cortical rotation. References. Sources: William K. Purvis, Gordon H. Oriens, David Sedava, H. Craig Heller, Life, The Science of Biology, pages 823–824. External links: micrograph of a primary oocyte and follicle of a monkey.